Hi, my name is Bala Chaudhry, and I'd like to talk to you today about some work that colleagues and I are doing to look at spore traits in arbuscular mycorrhizal fungi and how they might help us move toward a spore economic spectrum. So most plants on Earth in all ecosystems form symbioses with arbuscular mycorrhizal fungi. And these fungi don't form any above ground structures. They reproduce using multinucleate asexual spores that are born singly below ground. And these spores vary quite a bit with a suite of quantifiable traits. So they vary with respect to size, to surface ornamentation in the form of projections or even depressions. They vary with respect to wall thickness, shape, color, spore output, lots of different things that can affect the way AM fungi move, how they persist in the environment, and because these spores represent pretty substantial carbon sinks, how they interact with plant hosts. We've been using spore traits to help us predict aerial dispersal in AM fungi, and these are data from a paper that came out this year where we measured aerial AM fungal spores in the air and then measured them for a fair amount of traits. And so we found that aerial spores were mostly small, they lacked surface ornamentation, so they were smooth, and they ranged in color. So then we compared the traits of the spores that we collected to the traits of all AM fungi, all the Glomeromycota, with respect to size. And so in that comparison, we were able to show that about a third of described AM fungi form spores that are in the size range for aerial dispersal. So these fungi are thought to form mostly very large spores that can't disperse aerially, especially since they have no above ground structures. And so it was exciting to show that aerial dispersal may be much more prevalent for AM fungi than we previously thought. So spore traits can help inform for lots of other AM fungal life history strategies, not just dispersal, and they can also help inform on stability in the AM symbiosis. So Pierre-Luc Chagnon in 2013 presented this really nice trait-based framework for understanding AM fungal life history strategy. And depending on different AM fungal traits, species could be identified as competitors, as stress tolerators, as ruderals, and some of those traits are spore related, some are hyphal related, and then some relate to nutrient exchange capabilities. The problem is that there just isn't a lot of trait data for AM fungi. We have trait data for a handful of species, and people have been making comparisons between different families, Glomeraceae versus Gygosperaceae, but at the species level, we really don't have a whole lot of trait data available. So in this talk, I'm going to talk about a new database that we've been creating called Trait AM, which collates spore trait data for all the Glomeromycotina and includes an updated consensus phylogenetic tree. It's not completed, unfortunately, so I'm just going to share some of the data that we do have completed. And give an initial look at how spore traits can vary across AM fungal species, and we'll be looking at size, ornamentation, and investment in the protective wall of the spore. And we'll also look a little bit at how trade-offs might exist in investment in spore traits. And then finally, I wanted to just end with some new directions, so really new exciting questions that have been popping up with the ability to look at some of these traits across all AM fungal species, and how these functional traits, what the functional traits mean for biotrophic symbionts. So the database is made up of measurements of spores from original species descriptions. So we went into these species descriptions and pulled out measurements of on spore size and shape and wall structure and ornamentation. And it's not really something that could be mined, text mined in an automated way, because so many of these measurements, spore measurements, are carried out in different ways, and they're also reported in a huge amount of detail that the systematist made that we're not always necessarily interested in. And so it was quite a lot of work, and I want to recognize my student Emma Maras, who did a lot of it. And so we were able to pull out measurements of spores and then do some calculations. So for example, we have from spore diameter, we can calculate spore volume. And then from wall thickness, we can calculate the volume of the spherical shell of the spore. And then by looking at the ratio of those two, we can get an assessment of investment in a wall investment in spores. For ornamentation, we have heights of all of the projections, which would be a positive value. And then if the spore happened to have depressions, then we measured that ornamentation as a negative value. And then we recorded minimum, means, and maximum values for all measurements. So what do we see? How do spore traits vary across AM fungal species? So first, size varies quite a lot. So size varies by 4 orders of magnitude. And you can see we have spore volume split out by the 10 Glomeromycotina families. And indeed, there are some species that make very large spores, but then some species that make small spores. The Glomeraceae has a huge amount of size variation across it just in a single family. Other families are like Archaeosporiaceae, they have very small amount of variation. And I bring this up because we typically think of AM fungi as forming these huge spores, and they do. So those are the quintessential Gygosporiaceae spores. They exist on this end of the spectrum, but it's also important to remember that there's a lot of variation in spore size across different species of AM fungi. And just for comparison's sake, that the amount of variation in AM fungal spore size is similar to the amount of size variation that you see in eggs across all species of birds on Earth. Okay, what about ornamentation? Ornamentation was not as variable as size. So some species have significant ornamentation, some have none. But we did see a strong phylogenetic signal with ornamentation. So some families are more likely to contain species with ornamentation. And it was really interesting that ornamentations in terms of depressions, in the form of depressions, were only present in a single AM fungal family, the Acalosphoraceae. So that was kind of a new revelation to us. Okay, what about wall investment? So again, wall investment tells us what percentage of the total spore volume is made of the wall, made of the protective feature of the spore. And wall investment varies widely across AM fungal species. So on the left, we've got species that have very thin walls, so they invest a small amount in protective structure of the spore. And then on the right, we have some species that invest a huge amount in the wall of the spore. Almost 100% of the volume is the spore wall, which sounds odd to think about, but when you start looking at some of these species, so these are some of those high wall investment species, when we started to go back and say, well, what is this species? Here's a really good example. So this is a picture of Glomus nanolumens. That nanolumens literally meaning little with walls. And I mean, you can see how much of this spore is actually wall structure. So it's this little spore that invests a huge amount in the wall. It is the Helm's Deep of AM fungal spores. It's just a huge fortress protecting a small amount of resources. So it was really cool to see just this variation across AM fungi. Okay, now what about relationships between these traits? Are there trade-offs in investment in these spore traits? So we have a few predictions. So wall investment could remain proportional despite the size of the spore. So 50% of wall structure, 50% of volume is wall, no matter how large the size of the spore. You might also predict through allometric laws that investment actually increases as spore volume goes up. So this would predict that larger structures actually need a thicker wall in order to preserve the structural integrity of that cell. And then finally, we could see a trade-off where these traits, of course, are not free, and so the larger the spore, the smaller the wall investment, because there just aren't enough resources to go around. So what we found was data that supports a trade-off. So species with larger spores don't necessarily invest more in walls, and this pattern holds when we correct for phylogeny as well. So why is that? So why don't we see spore wall investment increasing and following these allometric rules? Well, we aren't quite sure, but we can speculate. And it could be related to wall chemistry. Maybe there are specific compounds in the wall that help cement that wall into place and keep it strong. Or it could be related to the ecology of the soil environment. And we can kind of take hints, going back to the bird analogy, from kiwi eggs. So kiwi birds form really large eggs with very thin, fragile shells, but they lay them below ground in protective burrows. And so the environment in where they're laid allows these eggs to kind of break the rules of allometry. And spores are the same. You know, they're born below ground in tiny pores of soil, and so it may be that their environment allows for a thinner wall with a larger spore. We've been having a lot of fun thinking about what these trade-offs might mean specifically for an obligate symbiont. So large spores are costly, thick walls are costly, but who is it costly for? Is it costly for the plant host? What does it mean for resource trade between the fungus and the plant? And we're excited to begin examining more traits and looking for the prevalence of other trade-offs and what it might mean for stability in the symbiosis or potentially cheating. We were really excited also about this ornamentation finding, particularly because we don't know what ornamentation does. Is it costly? What is its function? There's been speculation that ornamentation relates to animal dispersal, or maybe depressions are related to structural integrity. They kind of help hold the shape of this sphere, these rather large single cell spheres. We're not sure, but we are definitely excited to keep looking at more traits of the spores, and hopefully they can help unlock some of these spore economies, particularly if we start looking at other traits like color. Spore output is very important when we're looking at carbon costs to the plant. And then hyphal traits, of course, if we can figure out some way to incorporate hyphal traits, that would be excellent too, because it really is the main body of the fungus. So I'd like to just end by thanking our people, our funders, and thank you for listening and for tuning in.